you, and thank you for the organisers for giving me the opportunity to present this data today. It's a pleasure to be coming to you from beautiful Whadjuk Noongar land here in Perth and to be presenting some brand new data from our BLOSOM birth cohort. So the BLOSOM birth cohort was designed here in Perth to specifically study exclusively breastfeeding mother-infant pairs. So we had 85 infants enrolled in this study, and we followed them over the first 2 years of life. And as a microbial ecologist, I was incredibly excited to get my hands on more than 600 infant stool samples. Also, as a microbial ecologist, I did what we do best, which is to perform some amplicon sequencing. So in this study, we did full-length 6NS rRNA gene sequencing, which is, of course, a benefit over previous birth cohorts, which have predominantly relied on short amplicon sequencing. So sequencing the full-length 6NS gene gives a superior taxonomic resolution, largely to the species level. So without further ado, I'll dive straight into the results. This graph here is showing the composition of the infant gut microbiome over the first 12 months of life. For visual purposes here, I'm just displaying the OTUs that made up at least 0.5% relative abundance. So everything above the top of this graph here are these ultra-low abundance OTUs. And just off the back of the last talk, I know that it is quite difficult to differentiate various gradations of blue and green, so I've just highlighted some of the major features here. Our dominant taxa in this cohort was Bifidobacterium longum subspecies infantis, which I suppose would be expected in a breastfed cohort, and the second most dominant species was E. coli. I've also highlighted these two species in the bottom corner here. This is a Staphylococcus lugdunensis and a Streptococcus salivarius. And I thought this was quite interesting because these are two species that are very highly abundant in breast milk. So breast milk is predominantly made up of Staphylococcus and Streptococcus species. And I thought it was interesting that we had quite a reasonable abundance of these two species in the first week of life, but then they quite quickly tapered off, despite the fact that this was a breastfed cohort. But this actually lines up quite nicely with a hypothesis that's been put forward in the literature, which is that the infant gut microbiome begins as a facultative anaerobic environment. So the facultative anaerobes initially colonize, they consume the oxygen, and they make way for subsequent colonization by stricter anaerobes. So this data actually quite nicely supports that hypothesis. When we performed some beta diversity analysis, we identified 3 major phases in the gut microbiome. So this first phase here was a highly dynamic phase where each of these samples, 1 week, 1 month, and 2 months, was significantly different from all other samples. The gut then entered a highly stable phase where there were no differences in beta diversity from 2 to 6 months. And this was followed by another dynamic phase from 6 to 12 months. And 6 months, of course, is a very important time in the infant gut because this is a time where solid foods are being introduced. So, of course, these infants up until now have received nothing but breast milk. And then suddenly at 6 months of age, they begin to consume solid foods, which means that there's the introduction of a whole range of new substrates for bacterial metabolism. And we see that reflected in our alpha diversity measures here. So you can see we had relatively stable alpha diversity across the first 6 months of life. And then we had this very rapid uptick in both richness and Shannon diversity following the introduction of solid foods. However, even after the introduction of solid foods, Bifidobacteria remained as the dominant taxa in the gut microbiome. So across the entire cohort, almost half of our sequences mapped to Bifidobacteria. And this is likely due to the fact that Bifidobacteria are highly specialized HMO degraders. So as we've already heard today, HMOs, or human milk oligosaccharides, are a class of around 200 complex glycans. So these specialized sugars can't actually be digested by infants. So they arrive in the infant gut completely undigested, where they act specifically as prebiotics that have quite a bifidogenic effect. So there are bifidobacteria that can do internal digestion of HMOs and those that do external digestion. And regardless, we see that bifidobacteria are highly promoted by consumption of HMOs. And so even after these infants begin consuming solid foods, they still have quite a high abundance of bifidobacteria because we still had continued breastfeeding. So in our BLOSOM cohort, we've looked at a number of factors to see how they associate with the establishment of the gut microbiome. But in the interest of time today, I'm just going to focus on what I think is the most interesting story, which is that of delivery mode. In our cohort, about a third of the infants were delivered by caesarean section. And we know from the literature that there are two hallmarks of caesarean section delivery. The first is a delay in colonization with Bacteroides, and the second is a reduced colonization with Bifidobacteria. So when we look at those two hallmarks in our cohort, we see that it lines up nicely with the literature. So the turquoise bars here are our vaginally delivered infants, and the yellow bars are our caesarean delivered infants. And you can see that our caesarean delivered infants really lack Bacteroides until about the 12-month mark, where they begin to catch up to the vaginally delivered infants. When it comes to Bifidobacteria, the story is a bit different. So our caesarean delivered infants start with quite a low level of Bifidobacteria, but they quite rapidly catch up to the vaginally delivered infants. So by 3 months of age, there's no difference between the two groups. And this is likely because of the bifidogenic effect of breast milk, as we've already discussed. So it could be that the exclusive breastfeeding that we see in this cohort has allowed this rapid recovery of Bifidobacteria populations in our caesarean delivered infants. However, there's one confounding factor that we can't really disentangle from delivery mode, and that is intrapartum antibiotic prophylaxis. So in Australia, since about the early 1990s, if you are having a C-section, you'll be given intravenous cefazolin during your surgery. And this is to prevent potential incision site infections. So it's really difficult to tease out the effect of caesarean birth per se versus this routine use of antibiotics on mum. However, we do have a comparison group here. So about 20 to 30% of mothers who are delivering vaginally will also be given intravenous antibiotics during their delivery. So if you are colonized with group B streptococcus, or if you have prolonged rupture of your amniotic membranes, then you'll be given intravenous penicillin during your delivery. So in this cohort, we decided to compare these groups to see whether the antibiotic prophylaxis might explain what we're seeing. So here are our vaginally delivered infants. So in this case, the turquoise bars are the infants born to mothers who were not exposed to antibiotics, and the yellow bars are the vaginally delivered infants who were born to mothers who received intrapartum antibiotics. And you can see here a very similar effect in Bifidobacteria to what we saw in the caesarean delivered infants. And in fact, in this case, it takes them a little bit longer to catch up. So we're seeing this nice mirroring of that caesarean effect if we have a vaginally delivered infant who is exposed to intrapartum antibiotics. However, when we look at Bacteroides, the picture is a little bit different. So in this case, the infants born to mothers who received intrapartum antibiotics actually had higher levels of Bacteroides in their gut compared to those who were unexposed. And the reason for this difference could be due to the class of antibiotic used. So if you have a caesarean, as I said, you're given cefazolin, and this is effective against both Bifidobacteria and Bacteroides. And I've just listed Bacteroides fragilis here, since this was by far the most dominant species of Bacteroides in this cohort. And so you can see that the effect of this antibiotic maps very nicely to the effect that we saw in our cohort. Whereas if you're having a vaginal delivery, you'll be given penicillin. So penicillin is completely effective against Bifidobacteria, but Bacteroides are completely resistant to penicillin. And again, this maps beautifully to the results that we saw in our cohort. So our data suggests that when we mess with mum's microbiome during delivery, in this case by exposing her to intrapartum antibiotics, we can have knock-on effects for the infant. And so this is quite a significant finding given recent data that suggests that about 50 to 60% of the infant microbiome comes directly from mum. So regardless of delivery mode or mode of feeding, mum is the #1 donor of microbes to her infant. So when we expose mum to intrapartum antibiotics, it perturbs her microbiome, which means that she is then donating that perturbed microbiome to her infant in the critical months following birth. And this is really important because we know from several studies now that when infants are exposed to antibiotics in the first couple of years of life, it has lifelong health impacts. So to quickly summarise what I've told you today, the breastfed infant gut microbiome is highly dynamic across the first year of life and very strongly dominated by Bifidobacteria. Our data suggests that delivery mode alters the infant gut microbiome, potentially due to antibiotic prophylaxis. However, the good news story here is that we saw a rapid catch-up of Bifidobacteria colonisation in our breastfed caesarean-delivered infants. So hopefully that's a nice message that we can give to mothers who are nervous about having a caesarean section, that breastfeeding can help to rescue some of that phenotype in their infant. So with that, I'd like to acknowledge the fabulous group that I work in, and particularly those who were involved in the BLOSOM cohort. And I look forward to the Q&A.